Hi everyone. Um, Welcome to the channel. This is Dr. Asi, and today's topic is BSC 1010 lab laboratory and module one. Module one covers four labs: lab one, two, three, and four. Always remember our book, the orange book, BSC 1010 lab manual. So make sure that you check your notes, your lab experiments that you recorded, and then make sure to check the book. Okay, so let's get started. We have four labs again. Lab one was metric system, which is metric system and data quantification. So make sure that you look over the metric system and make sure that you can actually convert from non-metric to metric and vice versa. Also to know about the terms like data. Always remember data can be qualitative, which is something you don't count, but the quantitative like discrete or continuous. Continuous is data like a lot of data that has actually follows a normal distribution. So like your GPA, like gas prices, like so you can have any number, but in discrete, it's like having a full number, so not a or having colors or male, female, those type of or yes, no questions, right? So these are more discrete. Um, so we also measure a number of items there, and we made a graph. Make sure that how to how to make a graph and how to read a graph. Okay, so um, examples also like eye colors, Facebook likes, and those kind of stuff. Uh, they are more qualitative, but water temperature, drill depth, grades, these are more quantitative type of data. Okay, and also we talked about a hypothesis and then how we test the hypothesis. And in every experiment, we have negative control, which is basically the placebo, which should not give you the expected result because that's negative control. But it help us to see the effect of treatment group. So we should always have a negative control. So in many labs, we see negative control as water as our negative control, right? So, and one more item, specific gravity. So you should be able to calculate specific gravity, which is weight of a substance divided by its volume. So make sure that you're familiar with that, okay? So lab two, basically we see microscope, and lab three, cell. So lab two and three, they go together. Microscope, we talked about especially light microscopes in the lab and the parts. So make sure that you're familiar with all the parts of the microscope, how microscope works, how magnification works, right? Parts, magnification, what's wet mount, what's compound microscope. Even though we didn't use electron microscopes, but there are electron microscopes. And then after that, we looked at focusing on the microscope, making wet mount, and then move to the lab three, which was the cell. So remember, cells, we divide the cells in always into groups, prokaryotic versus eukaryotic. Now, prokaryotes, they don't have really nucleus, no membranes, membrane-bound organelles. Cells also can be unicellular or multicellular. And eukaryotic can be plant, animal, fungi. Plant cell has about 16, 17 organelles, but 3 of them are unique, or they are actually exclusively plant, like cell wall, chloroplast, and vacuole. So we look at onion cell, we look at Elodia leaf cell, and then we look at potato cells. So to draw and make sure that you know the differences and size-wise, you can be compare them. And then we look at also bacteria cell, much smaller cell. And now, what else you need to know in the cell? Basically, just to make sure that what dyes or assays we use iodine in some of them. Basically, we are going to use also in the next lab, and then make sure that you can identify where is chloroplast, where is mitochondria, where is nucleus, and where is a cell wall, and where is plasma membrane, those specifically, right? And then we see petri dishes, which basically we try to grow our bacteria in a petri dish with the agar media inside. So and put them in 37-degree incubator. So cells, cell parts, organelles, like 16 organelles, make sure, and then you can separate, or not only organelles, but the plant cells, different plant cells from each other, plant versus animal, like cheek cells, right? So, etc. Okay. Next lab is lab number 4. Basically, lab number 4 is chemistry of life. So we had a number of essays, which are also tests or techniques. So to do some food testing. Okay, so remember it's about macromolecules. Macromolecules are like carbohydrates, lipids, proteins, and also nucleic acids, right? We can have an essay or test for each of them, and also specifically like monosaccharides or polysaccharide, and like is a carb, lipids, protein, and also we see nucleic acid, and then we measure pH, right? So you use test tubes and always have a negative control, like water that should not change color, and then you had four maybe more test tubes, and then we had different food items in them, and always we needed high temperatures heating carbohydrates, but we never heat protein or lipids. So because they denature, so don't forget, proteins can denature if you heat them, so they get damaged. We use assays like Benedict, which turn orange color, or we use iodine, which turn black color. We use burette, like copper sulfate and sodium hydroxide combination, which turn purple or protein, like blue, purple, blue, violet. And also nucleic acids like DNA, we use ditch solution, and then it's also purple. And pH always actually goes from 1 to 14, and 7 is neutral. We use pH paper or pH meter to actually get kind of measure and assess the pH levels. If you're just interested in is it basic or acidic, we can also look at those color changes like blue or pink, and we can actually test different food items for their pH. So make sure you know the assay types, you know the pH scale, you know acidic, basic, you know these color changes, which each of them gives as a positive response. So these are lab 1 to 4, and this is the end of the review module 1, BSC1010. Thanks for watching, and please feel free to comment, like, and subscribe to the channel, and then see you next time. Best of luck and success, best of success. Take care. Happy studying.